Um, Dr. Prasanna, I think Dr. Samira has joined with my link, so you will see her name also with my name. Huh? Yeah, I
Ah,
think we can
okay.
start.
That's fine. That's fine.
Yeah, yeah.
Samira, can you try and share your screen to see whether it is working quickly?
Wait. Beresan, I will share my screen in a few minutes, but for now you can continue.
Yeah, can you share? Just wanted to check before we start the
Okay.
meeting.
No, unless I'm going to share this one. Let's go test. Okay.
So, friends, each one of us have 15 minutes each. So, kindly stick to that time. Let's not exceed that time. And if you are exceeding at any time, then I may have to come on the video and show you.
I should
Okay,
have seen
I
how
have
to
to
do it
give you
from
some
here.
indication, but don't ignore me. Okay.
Checking
So
this.
my request to you is, please do finish in time. Okay, and so that we can have enough Q&A session also. Otherwise, it will be simply we
Yeah,
speaking and people not able to ask any questions.
sure, sure.
So yeah,
Um,
kindly
I
move
think
fast.
Siaka has
Don't try
already
to.
shared.
Sorry, go ahead.
I think Siaka has already shared the screen, so maybe he wants to stop, so Samira can quickly check. Yeah,
No, no, you can just replace the current this
screen
thing.
straight away.
No, you can
Ah, okay,
simply
fine.
click on the current this thing.
Okay, okay, fine. I just want to just a minute. Uh,
Okay, the number is increasing, Sunny, almost 87 now, participants. So just let's give 1 minute time.
Yep. We'll just wait for Dr. Samira to try sharing her screen.
Yeah, Samira, can you
um,
please?
Dr.
Or
Prasanna, I think
Samira, if
she
you
has
can
some.
go to Supi's room and then use one screen, and that's another way you can do it.
Dr. Prasanna, I think she has some challenges. She'll join as we have the presentation, and she'll also share a screen. Then, yeah, yeah,
Okay.
Okay, then should we start
yeah.
then?
We can just, yeah, we can just start. Yeah.
Okay.
All right. We'll just wait for our team to have this webinar live stream on our Facebook. Then I'll start immediately. Thank you. All right. Hello, everybody. Good afternoon, good morning, or good evening, wherever you are tuning in right now. Welcome to our online learning event for today, February 19. This is the 54th webinar of the SEARCA Online Learning and Virtual Engagements, or SOLVE. This webinar is brought to you in collaboration with CIMMYT, or the International Maize and Wheat Improvement Center. I am Sunny from SEARCA, and I'll be your moderator for today's webinar. Together with our webinar training convener, we have Dr. B.M. Prasanna, CIMMYT's Distinguished Scientist and Regional Director for Asia. Our webinar's theme is Eco-Friendly Management of Fall Armyworm in the Maize-based cropping systems of Southeast Asia. And we're very happy to share that at this point, we see over 100 attendees from across Southeast Asia and beyond. For your information, this will be our program flow. After some introductions and reminders, Dr. Prasanna will give us an overview and objectives of this webinar. Then we will hear immediately from our resource speakers. There will be an open forum after all presentations for our attendees to ask relevant questions about their topics. After that, we request our audience for feedback through our evaluation form, and then we formally close the webinar. So some reminders for our attendees. If you are tuned in via SEARCA's Facebook page, you may type your questions in the comment section. Please include your location or country of origin. Or if you are registered and tuned in via Zoom, please post your questions or comments in the Q&A section located at the bottom or top of your screen. Please also include your location or country of origin as well. You can also type in your questions at any moment or while the resource speakers are presenting, and we will try to gather them and facilitate during the open forum. Alternatively, our speakers may also respond directly to your Q&As via Zoom immediately after their respective presentations. So please do not hesitate to post any of your questions or comments to their presentations. For you to access the recording of this webinar, it will be made available on SEARCA's Facebook page and YouTube channel. Today's presentation slides will also be available through our website at www.searca.org. You may also check our previous webinars from our website. And then that's it. All right, so now I turn this over to Dr. Prasanna to give us an introduction and the objectives of this training webinar. Sir?
Thank you. Thank you so much, Sunny, and a very, very warm welcome to all the participants of this webinar on eco-friendly management of fall armyworm in the maize-based cropping systems of Southeast Asia. I am Dr. Prasanna Buddupalli. I serve as CIMMYT's Distinguished Scientist and Regional Director for Asia. And before that particular function, I served as CIMMYT's Global Maize Program Director for almost 15 years, based in Nairobi, Kenya, but looking after the maize programs implementation in Sub-Saharan Africa, Asia, and Latin America. Today's topic of discussion is about fall armyworm, a pest that most colleagues have not been so familiar before 2018 or so in Asia. And even in Sub-Saharan Africa, before 2016, it's a largely unheard insect pest. But however, this pest has been there in the Americas for several decades, and that's how in the typical fall conditions, it used to appear in the corn belt, and when the temperatures go down drastically, the pest typically used to migrate to Mexico, Florida, and Texas, and further down into the Central America. That's the reason why this particular armyworm, Spodoptera frugipoda, was first called as fall armyworm. Since 2016, the pest made its appearance in Africa, and within a span of 1.5 years, it was reported in almost what we call 44 different countries. Since 2018, it made its foray into Asia, and by 2021-22, it moved almost all the way up to New Zealand and even Papua New Guinea and all these countries. So therefore, it's a ubiquitous pest now. It's there all over the world, except in a few parts of Europe where it has not been reported, or even if it is there, detected and destroyed at a very early stage. But right now, most parts of the global south have been impacted by the pest. And therefore, there is no single solution. There is no single organization that can provide solutions to this devastating transboundary pest that particularly likes maize. And it is highly prevalent in the maize-based agri-food systems. That's the reason why we partnered with the SEARCA. We are very honored to have this partnership with SEARCA and to conduct this joint training webinar on eco-friendly management of fall armyworm. What do we need to do? What are the various options available to us? Since 2016-18, there have been immense international efforts considering the concern that it has raised. A range of technologies and approaches have been identified by diverse international and national institutions. What is really important is how best to integrate these technologies and make them synergistic, make them cost effective, make them efficacious, make them safe to the smallholders who apply them, and make them scalable. So when we are talking about farm management technologies or approaches, we are considering 5 specific criteria: cost, efficacy, safety, accessibility, and scalability. So considering all this, the eco-friendly management is the key word here. We cannot continue to rely only on chemical pesticides to control fall armyworm and cause harm to the environment. So today, if you see the topics that will be covered by international experts, starting from Dr. Syed Nurul Alam from Bangladesh, then Samira from ISIPE, International Center for Insect Physiology and Ecology in Nairobi, Kenya, and then Dr. Subi Subramanian, Dr. Sevagan Subramanian, we call him Subi. Subi will talk about biopesticides for fall armyworm management, and then we go into host plant resistance, and finally into agroecological based approaches for fall armyworm management. So this is therefore covering about non-chemical interventions and how best we can monitor, scout for the pest, and what kind of approaches we need to take for eco-friendly management of fall armyworm. I do hope that you will benefit out of this webinar. These experts are indeed really world-class experts with excellent understanding, both theory and in practice, and working on practical solutions against this pest for over now 9 to 10 years. And therefore, what you get is a distilled wisdom out of these experts. So that's it from my side. Looking forward to a very productive webinar. Thank you.
Thank you very much, Dr. Prasanna, for that introduction. I'm sure our attendees at this point are now excited to hear from our international experts, as you said. Okay, so allow me to briefly introduce our first speaker for today. We have Dr. Said Nurul Alam. He is a distinguished entomologist with nearly 40 years of experience in research and development on integrated pest management. He has served extensively at the Bangladesh Agricultural Research Institute and with several leading international research and development organizations, including CIMMYT, IRI, AVRDC, and FAO. Dr. Alam is widely recognized as a pioneer of biorational pest management for major crops in Bangladesh, advancing sustainable and eco-friendly pest control practices. Ladies and gentlemen, let's welcome our first speaker, Dr. Said Nurul Alam.
Thank you, Sunny. I'm hearable. So, good afternoon and also good evening. Already Dr. Prasanna has already told about fall armyworm, some of the introductory things. Now I'm going to tell you some of in details about how to identify this polar armyworm, their biology, scouting, and monitoring system. So, you know that Spodoptera fujipadda, it's a armyworm. There are some other armyworms also, but this is a unique characteristics armyworm. Why? That I will tell later on. Already you know it's native to America. And now polar armyworm is a global threat for food production. And also, why? Because you know that the food security, food production, and rural livelihood. Because there are several regions, but main region is the polyphagous insect. Dr. Poshanno already told this maize, and maize is one of the most preferable crops for this polar armyworm, along with 80 other crops. Although there are some literature recently coming that more than 300 crops can infest polar armyworm, but among the 80 crops, the sorghum, millet, sugarcane, several vegetable, cotton, even rice in some area, wheat also can be infested with this polar armyworm. Till 2016, this was their home, native to America. And in 2016, first time, it was actually come to Africa. And then after 2016, it became going to almost everywhere in the world, except in some of the North Hemisphere. So it is actually infecting or impacting more in sub-Saharan Africa at this moment, Middle East, Asia, and Australia, even up to New Zealand. Already Dr. Poshanno mentioned. It is considered as a highly destructive pest for maize and other crops due to several reasons. Number one, migratory behavior. I will show you later why they are actually have this migratory behavior. It can capable of travel several hundreds of kilometers, the adults, overnight. With a wide host range, as I already told you, more than 80, although maize is the favorable one. Large range of environment and habitat suitability, and high reproductive rates. Come to its biology identifications. These are the eggs. Eggs laid on the leaves, especially in a cluster. But they are telling that we are calling it that is a high reproductive capability. Why? Because it can lay eggs 1,500 to 2,000. It's a unique one. Generally, this insect can lay eggs 200, 300 like this in adult female. So it looks moldy due to the depositions of scales. You see there are the scales between the and over the eggs. To save it from the natural enemies and all those things. This is all about eggs. I will show you some of the pictures later. Larvae. This is the neonate larvae. Feed starts just after hatching from the eggs. They start on the same leaves. The larva goes into 6 different instars, which is very, very much important because during this larval stage, the main infestation is going on. So first instar mainly aggregates on leaves. Second, third instars is in a dispersal mode. 4 to 6 instars is go inside the hole. But if you open the hole, you will see only one larva there, not more than one larva in one hole, because during these 4 to 6 instars, they have a cannibalism mechanism because they don't allow anymore. Generally, there are literature shows that around 40% of these pests fighting each other and is dying. Even after that, it became a dangerous and destructive pest. Pupa coming to them, this undergo inside the soil, making a cocoon and making silk. Adults, there are male and females, and they are active during the night times. These are all nocturnal. In daytime, it's very difficult to see in the field. Now, developing period, it's depends. Because I will show you later more that it has a temperature effect. Mainly, it's on its developing period. If the temperature is around 25 degrees Celsius, it's an average temperature. I'm telling that within 2 weeks, from egg hatch to 6-instar larva is actually completed. But about the larval behavior, it is very, very much important. I'm again mentioning that the larva is the main destructive time for its life cycle. First to third instar larva, we are calling it the small ones. During that time, they are not doing so much harm because the first stage of caterpillars actually need a little food. But when they are going to the whole 4 to 6 instar, they became voracious monster. These are especially these 4, 5th, and 6th. Voracious monster, and they can actually consume lots within 1 or 2 nights. 50% more they need than first, second, third instar or the small one period. Several special characteristics I am going to tell you. That's the overwintering limits, because they are from America. As you know, up to 2016, they were American. America is a almost temperate country. And during winter, this is under freezing condition. Unfortunately, they can't diapers, or they can't live, all they die under freezing condition. But if it is in a tropical and subtropical weathers, where temperature is stray above this freezing, they can actually, especially more than 13 degrees Celsius, they can survive. Their activity is ongoing. In the US, how then this pest became there for decades, year after year? Because in some of the area like South Florida or Texas, they can overwinter. And from there, they migrate. Problem is that if there is this temperature of 13.8 degrees Celsius, we are calling it a critical temperature. And when it's the temperature going below, all life stages, egg, larva, pupa, and adults are usually actually this stop. They cannot actually doing and after beyond this temperature. Migration strategy. It is a unique strategy of this pest because, and due to this region, they are actually surviving in the temperate region of this American continent year after year. During the summer or spring, where they were actually overwinter in South Florida or Texas, they start breeding more, and they migrate, reinfest northern latitude, even up to the Canada, and these places they go. Unfortunately, in tropical and subtropical weathers, they have a continuous life cycle. That is, they have the several generations in a year. Like in Bangladesh, we have seen 5 to 6 generations they can complete even in the maize crops. There is an overlapping generation. If you go to the field, you will see at a time these eggs, larvae, pupa, and adults. How to recognize? Number one, we can recognize through its life stages. The first sign, egg masses, as I already show you. And you see that the egg masses, they lay eggs 1,500 to 2,000, but not at a time, in a cluster of 100 to 150 like this, and in a scattered in some areas. Neonates come out and start feeding on the same leaves. Very important parts of their life cycle is their caterpillar or larva. There are the larval feeding sign, which is on which we can actually came to know that what's the status of fall armyworm in my field. So there are two types we are calling it. One is the early nesters, 1 to 3. We are calling them the small worms, and 4 to 6, which is entering inside the hole. We have to distinguish them, recognize them through their feeding signs. 1, 2, 3, or 2, especially the nesters, they are actually feeding on just the green parts of the leaves. So there is just like if you see the leaves against the sun, you can see the window panes like thing, only the veins of the leaves. But when it's go inside the hole, it's come out during the night time, and you will see the lots of frases. It is a voracious monster eating a lot, 4 to 6 nesters. At the same time, lots of frases is coming out from them. This is the hole feeding sign. You will see these types of continuous holes. How it's happened? I'm showing you these videos. You see how when the leaves are not in a fold, so they go inside during this fold from this part to that part. And in that way, when these folded leaves open, so you can see these types of these parallel holes there. So this is coming out from this. They are actually doing dig, and you see within these times, some frases they already done. So they are eating more, and lots of frases, fresh frases you can see. Main things for identification is the 4 dots on the 8th segment of the abdomen. This is a unique thing in especially after 3rd instar to 6th instar larva. And also there are 2 more longitudinal dark lines, and their mouth parts. We have to show the net-like structures. And this pupa goes take place in the soil at a depth of 2 to 8 centimeters. This is the pre-pupal stage. You see, they go inside the soils and start becoming ready for this pupation. This is the pupa. This is the adults. The male, you can distinguish very easily. We have to distinguish male because for monitoring, we are using pheromone traps, and for pheromone traps, we are catching the males. So this is the male, where actually there is this whitish marks on the tip of the forewings near the centers. But female, it is uniform grayish-brown. So there is a
Dr. Alam, 2 more minutes, please.
Okay. So this is for scouting and monitoring. How to do this? This is the for monitoring and scouting. We have to see why, when, what, and how we are doing this monitoring. Main goal is to detect the fall armyworm presence. Scout and monitoring is from the crop initial stage we have to do. Monitoring we are just doing with the pheromone traps, and scouting only on the larval feeding sites. This is the monitoring. These are the traps and these are the lures. So we are doing these 3 traps. We are setting at a distance of 100 feet away from one another, and traps are set after the seed sowing. It should be regularly interval after 1 week. Every week we have to see and the pheromone lures must be replaced at a monthly interval. And fall armyworm, this what it actually tell you. If one moth per trap per day, if you can see, then this is the threshold, because it is a high reproductive capability. One female can lay 1,500 to 2,000 eggs. Now coming to the scouting, because scouting and monitoring results actually give us a management decision. For that, we need a larval stage and plain growth stage. For that, sign of feeding is needed. These are the signs of, as I told you, the small worms. That is the small worm signing, and we are calling it SFW, small fresh window queen. These are for 4 to 6 large worms, signing of feedings. And these are the, in the fields, if these signs avail, we can actually tell them that these are the signs for large worms. But these times it is difficult for kill them because they are protective in the hole. So physiologically they are resistant. Third one is this infested plant. When they are actually going inside the cobs, this is the actually when the tassel is coming out, all the FAW larvae within this tassel or within this hole, they are actually coming out and attacking the cobs. So main thing is that the fall armyworm enters into the cobs, we must have to take care. If it is go inside, then it is very difficult to control. Mate growth stage is another for scouting. There are 4 stages, generally 3 stages we are calling seedling stage. That is this leaf number to 6 leaf numbers. We are calling in V1 to V7. Then 8 leaves to 12 leaves we are calling to mature stage. Then tassel to cob stage we are calling the reproductive stage. Here, although we are seeing 5 leaves, but actually there are 3 leaves because these 3 leaves has full leaves having the collars. So in that way, we have to count the leaves. And mature stage is not so much vulnerable because during that time, the leaves are actually coarse. So during that time, the insect cannot eat better. From where we have to get the data for SFW or IW? It is from the only new fully 3 open leaves. 1, 2, 3, not from the older leaves. How we will do the scouting? There is 2 methods. For the smaller plants, we have to go for W patterns. And for taller plants, we have to go for these ladder methods. What is that? These are the 5 points. From each 5 points, we have to select 10 plants in lines. And from those 10 plants, we have to keep records of the SFW for the small ones, IW for our big ones, that is the 4 to 6 insters. And if it is reproductive stage, then number of this cob infested. These are the in solar plants, which is actually difficult to go in this zigzag way. Then the same things, there are 5 points we have to see, and 50 plants from our scouting we have to select. Threshold calculation for SFW, 5 points, 50 plants, and from there we can have to calculate the percentage, like 12%. Here 50 plants, 6 infested for SFW, so it is 12%. Here these 50 plants, 3 plants for IW or a larger worm, so it is 6% infestation. During reproductive, one more things we have to see, that is the infested plant, that means the cob infestation. Here you see there's 50 plants, 8 infested, so it is 16% infested. how we will put this data. So this will have to format like this. Like in Los Banos Laguna, we have actually maize planted on 15th February, this last 15th February. So within 15 days, we have to start our monitoring and especially scouting. Maybe on 15 this March too, first this scouting we will done. That is at 15 days, the growth stage, maybe D1 or 2. And the window panes, SFW, maybe 6%, IW 24%, IW maybe 0, and infested plant maybe 0. After 21 days, again we will go, maybe on
Dr.
6
Alam,
days.
we need to wrap up, please.
So I'm just finishing because I have lots of 4 things. So that's why 3 more slides. Okay, please. So these have to 5 steps we have to repeat. That's the regional moth count before going to the field with the report. And for before field, we have to pheromone traps. 1, this male insect per trap per day is the threshold. Scouting, as I already told you. And based on this, we have to see. These are all the threshold limits, especially seedling. It must be more than 20%, SFW, IW. Mature, it is 50% because during that time leaves coarser. But in the productive, tassel stage is the more and more this dangerous. I'm telling you, this tassel stage. During that time, if the warm and dry weather, the control measure must be undertaken with most effective insecticide, pesticides, because during that time, if the infestation is high, the scab infestation will also be high later on. So last, the conclusion, I have already, it's an American pest now becoming almost a global pest, tropical, subtropical weather. It's a very dangerous due to its highly destructive, especially migratory behavior, wide host range, large environmental and habitat suitability, high reproductive rates. Effective timely management is very, very much essential for this with monitoring and scouting based. Few words about effective management. Don't rely on chemical pesticides because there are lots of biocontrol agents. So we must have to go for integrated pest management where with biopesticides and host plant resistance and agroecological management, which is actually following by these talks. So I am acknowledging these institutes and also Dr. Megra. And thank you. I'm just ready for your questions and your questions and now and also at any time. Thank you very much.
Thank you very much, Dr. Alam, for your presentation. As mentioned, feel free to type in your questions in our Zoom Q&A section. Okay, now we proceed to our next resource speaker. Allow me to briefly introduce him. We have with us Dr. Sevgan Subramanian. He is a principal scientist at the International Center of Insect Physiology and Ecology. He has over 19 years of research for development experience in Africa, and his significant research contributions are in the development of IPM strategies for horticultural and staple food crops, biopesticide research and development, climate change impacts and adaptation, and edible insects. Ladies and gentlemen, our second resource speaker, Dr. Subramanian Sevgan. Dr. Subi?
Subi, please put on your video.
Dr. Subi, you might be on mute.
Subi, you have to unmute and switch on your video.
Unmute myself and I'm switching on my video.
Yes.
And are you able to see my presentation now?
Yes, sir. You can try it on full screen, please. There you go. All good now, sir. Thank you.
Okay, okay. Thank you. First of all, thanks to Dr. Prasanna and also to our colleague from SEARCA for giving us this opportunity for making this presentation. I'll be talking about one of those IPM techniques which Dr. Alam presented previously, biopesticides for fallow management. And this presentation, most of the work that has been done is done from my side and also from my able colleague, Dr. Kombi Akutse. Just to go before a quick introduction about SCP, we are based in Africa and we are a center of excellence in Africa for insect science with over 50 years of history, with more than 571 staffs working with us. And we do a lot of capacity building for African scientists, 150 to 180 graduates trained annually, and we collaborate with more than 300 partners worldwide. We recently had our new vision and strategy brought up, and our vision is to transform livelihoods through insect science and innovations. And our mission is to deliver innovative, nature-positive insect science solutions for people and planet. I would appreciate if you have time, you can have a look at the vision to have an understanding of where recipe is looking forward to the space of insect science. To before quickly moving on to the topic, I guess Dr. Alam gave a very clear my thought process in terms of key points of intervention that we need to do for fallow management, especially the adults, which has the capability to migrate a lot. So they have to be targeted to prevent its migration. The egg stage, large number of eggs laid by the adults, and these egg stages have to be targeted by sustainable interventions. And also the larvae, which are the actual damage causing stage of this insect. And we need to work with the challenges that are there in terms of their concealing during the day, the economic damage that they have, and also the resistance to pesticides that these pest has got. The last stage, which normally does not take a lot of interest in terms of management widely, is the pupae, which is hidden in the soil. So we should also come up with strategies for managing this stage. So the whole idea is our management interventions should be holistic on all the different life stages to make sure that the efficacy is quite good. My talk will just not be restricted to the biopesticides, but I would like to expand it slightly to a broader scope of biorational pesticides, where the EPA definition clearly says biopesticides are certain types of pesticides derived from natural materials, animals, plants, bacteria, and other minerals. So within that scope, we have a lot of different things like the microbial pesticides, the plant-derived pesticides, and others, which I'll be taking you through with some of our work, largely from Africa, which is also, you can relate it to the Asian conditions as I speak. Why biopesticides? When you consider a parasitoid, it targets the egg stage and the larval stage. And when you consider host plant resistance, the target is mostly the larvae, to some extent on the eggs. But biopesticides has the potential to target all the different life stages which I mentioned. So there are fungus which you can target, which you can use to target the egg stage, larval stage, pupae, and the adults, all the 4 stages. The viral diseases targets mainly the young larval stages. There are recent reports on also its efficacy on some of the egg stages. And then we have the Bacillus thuringiensis, the BT, which targets mainly the larval stage there. You'll have this detailed information as I go through the presentation. So the idea is biopesticides are capable of targeting different life stages. So it has the potential to give a holistic control of fallow. When we start with the entomopathogenic fungus, these fungus are largely free living, and they have the potential to target the different life stages, as I said. They can be used both as augmentative control and also as conservation biology control efforts. Augmentative in the sense repeated application, while conservation in the sense conserving the ecosystem to have these entomopathogens work effectively to create epizootics. More recently, the role of entomopathogenic fungus, which normally also resides as colonizes plants as endophytes, is emerging. These are natural regulators of arthropods, and also as endophytes, they are non-pathogenic in most cases, and they also induce systemic resistance, which I'll be mentioning as I go forward. Yeah. Among the entomopathogenic fungus, there are two major groups of fungus which has been largely looked at in terms of management of lepidopteran pest, white muscardine disease, Beauveria bassana, which is effective against a different range of insects, termites, thrips, whiteflies, aphids, beetles, and all. With the Folamium also, the Beauveria bassina has entomopathogenic effect, especially against the adult more prominently as compared to the larvae and the eggs. The critical feature of this is in the early stages of its growth, the fungus will be pure white in color, which can be an indication that yes, you are having a Beauveria infection. The next one, which has largely been tried out in Folamium management, is the green muscardine disease, or the Metridium anomarea, which we call it so. Within this, Metridium anisophilae and also Metridium bruneum and Metridium rilli are ones that are now increasingly being seen on Folamium controlled space. So as with the Beauveria bassina, it is also widely seen in the nature, and it also has a lot of different hosts apart from Folamium as well. One of the difference between Beauveria and Metridium is Beauveria is more reported as endophytes, and it's more. Hello? Light break. Hello?
Subbi, we can't hear you.
Okay, there is a slight power drop. So are you able to hear me now?
Now we can hear.
So Beauveria is more known for its endophytic properties, while Metridium is not so much endophytic in nature. So these are some of our findings when we tried out the EPFs against grown-up larvae, Folamium. We didn't find a lot of promising efficacy, while the Folamium are highly susceptible at the very young stage of egg and newly emerged larval stages. And at TCP, we maintain a lot of different isolates of entomopathogenic fungus, both Beauveria and Metridium. And of this, we have found some interesting isolates which have already progressed into commercialization, which I'll be presenting. Yeah. These isolates, especially the ECP7 and 78, which showed promise against Folamium control, has now been tested widely in the field in East Africa. And we have also tested the efficacy of some of these isolates on the adults. Why adults? Because adults are capable of by targeting adults, you can reduce the fecundity of the adults, and also you can use the adults as a kind of a vector for the entomopathogenic fungus. So as I said, in the adult stages, Beauveria bassina has a good efficacy comparable with Metridium and other isolates that we have studied so far. Whenever we apply biopesticides, I think we should make an effort to reduce the cost of use of the biopesticides, and in this space, innovative application strategies can help a lot. Horizontal transmission of EPF inoculums from one moth to another during the mating process can be quite useful, and in this regard, we have studied how the fungus, when applied on a male, gets transferred to the female, or from the female to the male, and what kind of mortality is caused. So clearly, both as the male and the female as a donor, the recipient sex tends to have a good level of mortality. And the additional advantage that you have is when a female is inoculated with the fungus, either from another female or from another male, their ability to start laying extensively decreases quite drastically. So this reduces the fecundity. We tried also compatibility of entomopathogenic fungus with pheromones, and we are able to clearly find that yes, the entomopathogenic fungus are compatible with the pheromones. So this gives a clear opportunity of developing auto dissemination tools, where the tools can be used for both monitoring and also for as a focal point for disseminating entomopathogenic fungus into the Folamium population. This is what I was saying, that apart from spraying the entomopathogenic fungus, you can also try developing them as endophytes, clear endophytes, because the advantage of this is you have a very reduced cost of application of the fungus. Oops. Yeah. We have tried several endophytes, Trichoderma species, Atroidae herzianum, Hypocreolixi, Trichoderma asperellum, and others. And clearly, the number of egg masses laid when the plant is colonized with an endophyte decreases, and also the feeding potential also changes for Folamearum in an endophyte colonized plants. Are you able to hear me? Hello?
Yes, Dr. Subi, we can still hear you.
Yeah, thank you. Apart from the biopesticide being effective to the Folamearum, they should also be safe to the other biocontrol efforts that we have, especially the parasitoids. And in this regard, we have tested the safety of entomopathogenic fungus to parasitoids. And here is an example of Telenomus remus. And what we observed is Telenomus remus adults are not so infective to the entomopathogenic fungus. And also, when the parasitoid is allowed to parasitize an egg, and the entomopathogenic fungus is applied later, the emergence rates of the parasitoids are not very significantly impacted. So clearly, these two techniques could be integrated in an integrated pest management strategy for Folamearum. Following all these results, it is critical that it shouldn't remain in the lab. And in this regard, we have engaged with the regulatory authorities in East Africa right from 2019. And currently, the entomopathogenic fungus, both SCP-78 and 7, are registered in East Africa in 3 different countries: Tanzania, Kenya, and Uganda. And very recently, we also got it registered in Malawi for use in Folamearum management in these countries. The next biopesticide that has taken a lot of space in terms of Folamearum management is the nucleopolyhedrosis viruses, previously which was called as nucleopolyhedrosis virus. It's an obligate pathogen of Lepidopteran, so it can only survive on the host. The infection is through oral feeding, and early instas are very susceptible. Interesting enough, with Spodoptera frugipeda, these baculoviruses are highly specific. So normally in other situations, a baculovirus that is infective to one species will not be infective to the other species of the same genus. But interestingly enough, what we are observing from what literature has come out is it's not just Spodoptera frugipeda nucleopolyhedroviruses that are infective to Folamearum. Some strains of Spodoptera litura in Asia and some strains of Spodoptera litoralis in Africa are also capable of infecting Spodoptera frugipeda. This is because also the diversity of the virus that is found in the ecosystem. These are some of the products that are already available, and also some of them which has been tested in Africa: Foligen and also Litovir. Litovir is actually a Spodoptera litoralis virus which is infective against Folamearum, and Foligen is a Folamearum baculovirus. Clearly, the insect has to consume the virus, and it has a specific incubation period, so it is not very rapid in its kill. But the advantage with these viruses are they get transmitted through generations. So there is a kind of horizontal and vertical transmission, so can contribute to decrease in population over time in the field. In Africa, there is some interesting work done by our partners CABI with the national partners in testing the existing baculoviruses for Folamearum control, and also looking at opportunities to see how recycled virus. So, for instance, you spray a farm and you also get larvae from those farms which are infected with the virus, and you recycle this virus and try to spray, because mass production of this virus from commercial scale is costly. So this was tested in with the smallholders. The recycled virus are not as effective as the commercial ones, but still they are at times better than the untreated control, which can add to the Folamearum control strategies. The next group of biopesticides is the entomopathogenic nematodes. Clearly, the mode of action is they can infect through the spiracles and also the openings of the insect.
Last 2 minutes, please.
Quickly, I'll finish up. Yeah. So there are several nematodes that has been screened. Clearly, commercial nematodes, Stenodium carpocaspae, gives up to 100% mortality, while there are also other nematodes that has been observed in the wild, which needs to be developed in terms of production and formulation and this. Yeah. The advantage of the nematodes, it's not just infective to the larvae. Very recent information from India indicates that the nematodes can also be infective to the egg stage and also to the pupal stage, giving a broader scope for control of Armyworm with entomopathogenic nematodes. The last one is the Bacillus thuringiensis, gram-positive bacteria, which infects the gut and makes the insect not to consume a lot. There are several commercial products that are available for BT, but more and more BT is looked at as a transgenic crop rather than the product to control pest, which I think Dr. Prasanna will cover in his talk. Apart from the biopesticides, there are several botanical options that has been tested all over the world. It is unique to the context of the situation. Botanicals present in the local situations contributes to fall armyworm management. So a resource poor farmer can access some of these botanicals and also add to his repertoire in terms of fall armyworm management. This is a study that was done in Ethiopia where we found that neem and also Shinus mole and other botanicals can contribute to fall armyworm management or reduction in the larvae. The last one which I want to cover is biopesticide is not just about spraying a molecule for killing the insect. This is a traditional practice in Malawi where fish soup was said to be effective against fall armyworm. The soup is not toxic to the insect, but it recruits a whole lot of natural enemies into the system. And this is what we studied. And these natural enemies, predators, actually contributes to the biocontrol effort of fall armyworm. Yeah. So some of the advantages of biopesticides: it's ecologically safe, biodegradable, no harmful residues in the crop, often highly effective for management of pesticide-resistant insects such as fall armyworm, and target-specific and safe to natural enemies. Can be effectively integrated, but the challenges with the biopesticides are costly in comparison to pesticides. So we need to have improved and local production and formulation techniques. Another way of reducing the cost could be innovative application strategies, auto dissemination, and others. Weak regulatory frameworks is also a challenge. We need to create enabling policies to register these biopesticides quite quickly in the different parts of the world. So in this regard, we have engaged in a lot of global level trainings on biopesticide production and also biological control agent productions. And as my previous colleague clearly highlighted, biopesticides are just a part of the entire spectrum where active monitoring, use of biopesticide, resistant cultivars, which you'll be hearing from Dr. Prasanna, and also capacity building are critical in establishing a comprehensive package of practice for fall armyworm control. With that, I would like to conclude and thank some of our donors who have supported our work, and I'm happy to take any questions as it comes. Thank you.
Thank you very much, Dr. Sui, for the presentation. We now move on to our next resource speaker. We have with us Dr. Samira Abulgasi Mohammed, a Sudanese national. She is an agricultural entomologist and biological control specialist. Currently, she is a principal scientist at the head of the plant health theme at the International Center of Insect Physiology and Ecology. Her main area of interest are pre- and post-harvest management of native and invasive pests using agroecological based approach with main focus on biological control using parasitoid. Ladies and gentlemen, our next speaker, Dr. Samira Mohammed.
Samira, are you there?
Dr. Samira, would you like to start sharing your screen?
Samira, please share your screen. Okay.
Hi, Prasanna. Okay, we are trying to share, but we have also a technical problem. I think you can proceed with other presenters. I had a technical problem from this other end. Please continue other presenters. I will come last.
Okay, fine. So in that case, Sunny, I will go ahead.
All right. All right, sir. So we'll proceed with the next presentation. We have Dr. Prasanna again. Allow me to briefly introduce him. Dr. Prasanna is CIMMYT's Distinguished Scientist and Regional Director for Asia and Managing Director of the Berlog Institute for South Asia. He served as Director of the CIMMYT Global Maize Program from 2010 to 2024 and is internationally renowned for his contributions to the maize genetics, breeding, and deployment of hundreds of high-yielding, climate-resilient, and nutritionally enriched maize varieties for the rain-fed, stress-prone tropical environments in sub-Saharan Africa, South Asia, and Latin America through extensive partnerships. Ladies and gentlemen, our speaker, we have Dr. B.M. Prasanna. Sir?
Thank you very much, Sunny, and to the entire SEARCA team for this wonderful opportunity to provide the perspective on various eco-friendly approaches for fall armyworm management. What I will be presenting is on host plant resistance to fall armyworm, for which CIMMYT is highly renowned. The breeding for native genetic resistance to fall armyworm started way back in 1990s when Dr. John Mim, this gentleman here, a maize entomologist, started screening thousands of land races and breeding materials for their resistance to various insect pests. And through that work in 1990s, Mim and team identified that there are two most important sources for insect pest resistance in maize. One is the Cuban flints. Another is Tuxpeno, which is a kind of a landrace accession in Mexico. So there are two excellent publications that followed in 1997, the insect-resistant maize, recent advances and utilization, where the protocols for mass rearing of fall armyworm, the sources for native genetic resistance to fall armyworm have been highlighted. But when you look at breeding for native genetic resistance to insect pests, be it fall armyworm or any other insect pest, it requires good knowledge of the biology of the host as well as the pest. The effectiveness here undoubtedly depends upon what kind of diversity of germplasm that institution has. Does the institution have an efficient insect rearing technique? Do you have an efficient artificial infestation protocol of the host so that you can generate robust responses? And what kind of breeding technologies or methods that the institution is following to transfer the resistance once it is identified in any breeding material into elite genetic materials. When the fall armyworm broke up in 2016 in Africa, CIMMYT, at the time I was the maize program director, we established this world's largest facility for fall armyworm screening at our research station called Kibako, which is the 2.5 hours drive from Nairobi. At this site, you can see 16 to such screen houses, each screen house 1/3 of the size of an acre. And it can hold several, you can grow several thousand entries within that particular screen house, artificially infest each and every plant, and not only at the vegetative stage, but also at the early reproductive stage, and then identify materials that are native genetic resistant. So using MIMS materials as well as many others that are adapted to Africa, we started screening germplasm at our Kibako facilities. Over 20,000 germplasm entries were screened against fall armyworm under artificial infestation at the Kibako facility. We also established a similar facility in Hyderabad, not to the same number of screen houses, but about 8 screen houses were there, where Asia adopted CIMMYT maize germplasm have been systematically screened against fall armyworm under artificial infestation. You can see under such conditions, how does a susceptible inbred line look like compared to a resistant inbred line? The result of all these studies, right from 2017, we published in 2022 an article in Theoretical and Applied Genetics on host plant resistance for fall armyworm management in Asia. what's the relevant status and prospects for both Africa and Asia? One important thing that we need to do whenever you do germplasm screening for host plant resistance is not only take into consideration the foliar ratings. That means right from score 1, which is unaffected completely, no even pinholes on that, all the way up to score 9, which means it is completely damaged at the foliar level by the insect, severely damaged. Not only foliar responses, but also the ear responses need to be recorded under artificial infestation. Right from score 1, that means absolutely no effect, to score 9, where all the kernels were eaten away by the fall armyworm, because the insect can bore through the silk as well as through the ear husk and then cause extensive damage. This data recording is very critical. Don't look at only foliar data, but also the ear data. If you look at the foliar data, you can see CML 71, CML 338, CML 444. CML means CIMMYT maize line. CML 574, CML 338, CML 71 offer excellent even foliar responses under artificial infestation compared to, you can see here, a drought tolerant inbred line, which is highly vulnerable to fall armyworm, or a CML 395, which is an elite line. So using this robust protocols, we identified a set of inbred lines which are excellent against fall armyworm and distributed globally: Africa, Asia, Latin America, North America, even Europe, all the way up to Australia. These are the number of institutions, and these are the number of countries. So you can see 92 institutions from 34 different countries have benefited out of this germplasm. The second most important thing to do is, once you have a highly promising hybrid that is identified through choice trials, where hundreds of entries are grown within the screen house, you have to also set up a no choice trial. No choice trial means one big screen house divided into 2 or 3 different partition, and each of this chamber will be growing only one entry, and each and every plant in that chamber, that means even 30-40 rows of a hybrid, are infested at the foliar and reproductive stage, and then you record the response. That means there is no confounding effect of any other germplasm, any other entry. And under such conditions, how does your entries respond? Either the insect has to survive and eat, or it has to perish. So under such conditions, you can see here the fall armyworm tolerant CIMMYT hybrids absolutely clean in such no choice trials, compared to susceptible commercial checks which are severely defoliated. You will hardly get even less than 1 ton per hectare. And even if you get some grain, it will be highly contaminated with insect eating. On the other hand, you see here how the fall armyworm hybrids are performing. So, and then when you look at the yield data from those no choice trials, this is how clean are the ears compared to the insect eaten ears, if at all they are there. First of all, the yield is severely penalized. Second is, even if the yield is obtained, the grains are very much prone to susceptibility to various other fungal infections. So through this strategy, in December 2021, we released the first set of fall armyworm tolerant white maize hybrids for Africa. And now Kenya, Ethiopia, South Sudan, Zambia, Malawi, Ghana, and Zimbabwe have not only already released these hybrids, but they are now commercializing through seed companies. Similar national performance trials are ongoing in several countries in Asia. My request to the Philippines colleagues is to get this germplasm, evaluate it under local conditions, and start releasing fall armyworm tolerant native genetic resistant materials. So we have now, as I told, intensive screening being undertaken at CIMMYT Hyderabad under fall armyworm artificial infestations. Already a total of over 138 and inbred lines identified to be tolerant after rounds of screening. Over 30 breeding populations with fall armyworm resistant donors have been generated and used in our breeding programs. You can see here again our resistant hybrids from CIMMYT Asia pipeline compared to the susceptible commercial checks in some of our Asian countries. So it is really quite strong, almost 5 to 6 tons per hectare difference between a fall armyworm resistant hybrid versus a susceptible hybrid. The question that we need to ask next is, what causes this native genetic resistance? A range of characters, right from morphological traits, physiological traits, biochemical traits, and even molecular basis of native genetic resistance is increasingly being understood, including proteins related to, for example, cysteine protease, or beta caryophyllin. It's a terpenoid molecule or a secondary metabolite. So a number of genes, including those in terpenoid biosynthesis, flavonoid biosynthesis, have been identified. So it's not only non-preference, but also antibiosis that you see. Non-preference is because of leaf thickness, high trichome density, tight husk cover. Those are typically contributing to non-preference of those hybrids. And these secondary metabolites, including phenylpropanoids, caryophyllins, etc., they cause antibiosis. So we are now increasingly understanding the basis for native genetic resistance to fall armyworm and what inbred lines offer what sort of mechanism. For example, CML71 not only shows good antibiosis, but also good non-preference for feeding. Whereas CKSBL, that is a CIMMYT Kenya stem borer resistant line, showed excellent antibiosis on leaves, besides non-preference for feeding on leaves, silks, and ears. So several national partners have started using these materials in breeding for native genetic resistance to fall armyworm, including colleagues in Ghana, including colleagues in India, Zimbabwe, and the list is several countries. So I'm just showing you glimpses of this. Coming to the BT maize, there are, of course, BT maize projects which are implemented in Africa in collaboration with Bayer, with African Agricultural Technology Foundation in 5 different countries in Africa. BT maize has been already released in not only in South Africa, but also in Nigeria, and recent approvals are also coming up in Ethiopia. So this is what we call as the Taylor maize. It's a Monate 9034 BT maize, which can offer resistance to fall armyworm as well as stem borer. In Asian context, BT maize has been in cultivation in Philippines since 2003, in Vietnam since 2015, in China it has been recently released, in Pakistan they are being tested. So there are a number of countries in Asia too which are beginning to grow or give approvals for BT maize hybrids. But one thing one has to be very careful about when we are talking about BT maize is insect resistance management. Already we are seeing in China and in many other countries insecticide resistance characteristics in several of the fall armyworm populations that are invading this country. So robust insect resistance management has to be a key component of an integrated pest management strategy, especially if you are relying on BT maize. You have to prevent or mitigate the onset of resistance. You have to better understand the population dynamics of fall armyworm and resistance profiles across the region to guide our response strategy. But if I have to manage fall armyworm, whether it is in Africa and Asia, in a much better way, I won't rely only upon BT maize. I will rely on native genetic resistance too, because native genetic resistance is a polygenic trait compared to a BT maize, which is typically monogenic or oligogenic nature. So together with Bruce Tabashnik, the guru of insect resistance management, we came up with an article on how can we sustainably improve control of fall armyworm. So where we recommended clearly use BT maize with other pest management methods, including native genetic resistance as a part of your IPM plan. Avoid single toxin BT crops. Go for BT maize pyramids and things like this. So I would certainly prefer a BT gene to be incorporated within a native genetic resistant background rather than a susceptible background. So the resistance allele frequency of fall armyworm are changing for CRY1, CRY2, VIP3A. There are patterns of cross resistance. There are fitness costs associated with resistance. So that's the reason why new BT toxins are also being probed by several institutions within Asia. One more aspect is genome editing for fall armyworm management. The fall armyworm Spodoptera frugiperda has been sequenced. The genome has been sequenced. Genes responsible for various critical reproductive functions have been now identified in fall armyworm. So this would provide an opportunity to create fall armyworm populations it's like for providing mating disruption and release that in environments. That could be one potential strategy. A number of such functional genes have been characterized in Spodoptera frugiperda in recent years. Look at this paper in 2024. An array of genes have been identified affecting different aspects: chemosensory regulation, recognition, biosynthesis of female sex hormones, pheromones, sex determination and reproduction, growth and development, spermatogenesis. There are a number of gene functions that have been identified. So in summary, friends, both conventionally derived and BT-based resistant maize varieties have their own importance in fall armyworm management. We do need to intensify breeding activities in South and Southeast Asia for developing elite maize germplasm with fall armyworm resistance and fast track the deployment of fall armyworm tolerant elite maize hybrids. In countries like Philippines, where BT maize deployment is happening, one must give due attention to insect resistance management. And it is certainly possible to achieve synergies between host plant resistance and other IPM approaches for eco-friendly management of fall armyworm. Thank you so much.
Thank you very much for the presentation, Dr. Prasanna. Should we head to Dr. Christian
Sure.
or okay? So Dr. Samira will be the last presenter. So let's proceed to our fourth speaker. We have with us Dr. Christian Tierfelder. He is a principal cropping systems agronomist at CIMMYT, specializing in conservation agriculture or CA systems research across Southern Africa. He led CA research and implementation projects in Malawi, Mozambique, Zambia, Zimbabwe, and Namibia since 2004. His work focuses on adapting CA systems, cultural practices, and appropriate scale mechanization to the conditions of smallholder farmers. Ladies and gentlemen, let's welcome Dr. Christian Tierfelder.
Hello, everybody. Thank you so much. Can you hear me properly?
Yes, sir. Loud and clear.
Wonderful. Then let me continue with my presentation. I'm not a fall armyworm specialist, I must say, but I specialize in cropping systems research. And that has a good reason in this presentation. So when we observed in 2016-17 the emergence of cropping issues with fall armyworm, as you can see from this map, we were puzzled by differences in cropping systems, how they responded to fall armyworm. And that was an interesting aspect that we wanted to explore further. Because, for most smallholder farmers that I'm dealing with, one of those very highly sophisticated control options, BT, are probably out of reach. But cropping systems, if combined with, for example, resistant or tolerant maize varieties, could be a combined control strategy. So when the pest arrived in this area, the immediate reaction by governments was chemical control. They panicked. They all bought pesticides, often insecticides that did not even work for fall armyworm and were not actually released for that purpose. So some of the responses were not very encouraging. The damage was horrible in some areas, and some of the yield penalties that were portrayed by several researchers were enormous. Yield losses between 20 and almost 70% were postulated, but the reality is actually slightly different. Control methods based on agronomic strategies are probably more affordable for resource-constrained smallholders. They represent low risk for health and the environment and can be easily adopted. So our objectives in this work was, first of all, to estimate fall armyworm damage in smallholder maize fields outside research stations with real smallholder farmers in the field, following these rigorous scouting protocols. Then understand factors influencing the fall armyworm damage, identify natural enemies, and also identify different cropping systems that could help controlling fall armyworm. And we're building on the knowledge from organizations like ICIBE, for example, that helped us really design climate-adapted systems for our situation. And that was mostly Southern Africa. So what we did in the first step, we did some field assessment in rural districts of Zimbabwe in 2 cropping seasons to really understand the damage, severity, etc., of fall armyworm in smallholder farmers' fields. We use those zigzag protocols that have been shown before, looked at number of plants with leaf damage, number of plants with frass in the world, damage scores that have been also looked at, and also scores on the crop itself. We then used a technique that CIMMYT developed over the years for a rapid yield assessment, looking at crops that we harvested from specific areas that we photographed, used a computer to evaluate how much yield we would get from those using specific techniques developed by my colleagues from the maize program, and then assessed what would be the yield of those systems. So what we found out in those 2 years where we did this research in 2 different districts, Chipinge and Makoni, that we had leaf damage in the range of 42 to 55% in the first year, and then 75 to 49% in the second year, and damage scores around 3.7, 3.8, sometimes a bit more, 5.3 in the second year. And then what was very interesting, we then looked at factors influencing fall armyworm damage through a very thorough survey that we did with the farmers, asking them about their cultural practices, what they do in their fields. We found, interestingly, very little effect of those pesticides that were applied at that time. Now we have better pesticides that can control, but at that time, no effect. Even the presence of hedgerows or pumpkin intercropping did not lead to any positive effect on fall armyworm. Instead, for example, pumpkin leaves could be a hiding place for the moths or hedgerows from where they migrate into the maize stands. And some maize varieties showed a little bit of control, some absolutely not. And that's actually when we become very, very grateful for the work that Prasanna and his team did. Then rotations with legumes or fallows were very beneficial, especially with legumes. Conservation agriculture, a cropping system based on minimum soil disturbance, crop residue retention, and crop diversification was very effective. A balanced fertilization was good. Application of composts, of frequent weeding, or early planting were medium to well in controlling fall armyworm damage. We also tried to estimate the actual yield loss that we would get with different infestations. And what we found in those 2 years, only some 12% to 22% yield loss from the actual work. And when we just looked at the crops themselves, it was slightly lower. So those huge numbers that were initially put forward did not apply for our situation. That could be several reasons, but this was not as dramatic as everybody thought it would be. We then followed on to establish on-farm trials, replicated on-farm trials with different, what we call climate-adapted push-pull systems. Push-pull is a system that was developed by ICP. I'll talk about that in a minute. So we used crops that are common in the farming systems of Southern Africa and intercropped them with maize, with brachiaria hedgerows and without brachiaria hedgerows. And then we looked at what would be the effect of this. The push-pull system is a system basically developing two control mechanisms. One is this push that comes from a legume called Desmodium unsicatum, and the pull from a grass that attracts the fall armyworm out of the maize stand and helps with this double action of push and pull to control the pest. We interestingly found several legumes that reduced the damage. For example, cowpeas as an intercrop, or lablab or velvet bean, even groundnuts reduced the damage scores in the field. And that's actually what we wanted. We wanted crops that are available and are used by farmers in the fields to help us control fall armyworm. We also had traps in the field to really find the enemies or the positive insects that help us control fall armyworm. And it was interesting. Spiders, parasitoids, ants were measured in those two districts, and there were differences as well. In some districts, for example, ants were much more pronounced than in other districts. And some of the cropping systems also responded better to those, and we found more of them. So that made us think, what is actually helping us in controlling fall armyworm and what is actually not helping us. So ants were identified as one agent or one insect that really helped us in reducing the damage. The more ants we had, the better it was for the maize. Also parasitoids, although we did not establish such a clear relationship with those. It's a complex issue. What you need is animals, insects that attract, for example, the natural enemies. Here we have the alternative prey. These are insects that are good for attracting ants. And then we have the enemies. In this case, formicid is ants. The more ants you have, the more we can actually increase the predation of the fall armyworm, which then reduces the damage. Some crops also had very strong volatiles, like in this case, velvet bean or mucuna, a green manure cover crop that was very strong in repelling the fall armyworm, that also reduced the fall armyworm damage. So, and then lastly, we also had a specific situation in Southern Africa. We have a 7-month dry period from about May to November. And farmers grow maize under irrigation in such areas and really suffered from fall armyworm because there's nothing else green. The fall armyworm goes for those plantations, for those green maize production areas. And they really said, well, we have damages of 50, 80% in our maize and we can't sell our green maize anymore. So what we did, we put a few lures in those and basically trapped all fall armyworm females in those. And where we did that, we could control the damage in those plantations. And that's probably a strategy as well that I mean our colleague from ICIPE has now talked about. So in conclusion, several agronomic practices appear to influence fall armyworm infestation in smallholder conditions. Legume intercropping or strip cropping has been very beneficial. Conservation agriculture has been super effective. And organic amendments as well, because they increase the abundance of natural enemies. Those natural enemies, like the ants that I was talking about, help naturally control the fall armyworm. Some maize varieties appear to be more susceptible. We learned that from Prasanna no more in detail. Now we are a few years later. We have these tolerant varieties now that help, of course, in reducing. So combinations of the cultural practices and the good new elite varieties will really help us reduce those damages. And these technologies are in reach of smallholder farmers. There is a certain effect of seasonality. Some years were very dry, some very wet, and we found that this really makes a huge difference for smallholder farming areas and farmers' fields. In drought years, we had huge damage problems in some areas. Early planting is important. Frequent weeding as well, because often grasses in the stand are of the same nature. They're also cereals, and that's why fall armyworm can continue breeding on those. So if you weed continuously and have clean fields, you can control fall armyworm. Then crop diversification, as I said, is in any form beneficial because it will increase the diversity of not only one type of environment in agroecology. It brings in a whole range of new insects of various nature. And if nature is in balance, this is the best way to control an environmental pest like fall armyworm. Then finally, yield losses are seasonal, with perhaps an interaction with dry season, late planting, and high damage and yield loss. But this is really dependent on the seasonal situation that you are in and what we can expect from different types of seasons. So in retrospect, 4 or 5 years after the emergence, or it's even more than that now, it's 9 years now, we have adapted to the situation. We are not there like we are in Latin America, that everything is in balance, but we have new technologies, we have new varieties, we have intercropping strategies, and together they can work very efficiently in combination to control this damage, the damage of the fall armyworm. Thank you so much.
Thanks for your presentation, Dr. Christian, for sharing with us those interesting insights from your research. Okay, now we can proceed with Dr. Samir's presentation. Dr. Samir, are you able now to share your screen?
Presentation itself, it got corrupted. So now we are sharing.
Yes, we can hear you, Dr. Samir. You can try sharing your
Yes.
screen. Thank
Yeah,
you.
we're sharing. Okay. You can see my presentation?
Yeah. Go ahead.
Okay.
Can you try it in full screen, please?
Yeah, yeah, good. Go ahead.
Yeah, all right. Thank you.
Okay. Thank you so much. Thanks, Prasanna, for inviting me for this webinar. Thanks so much appreciated. And sorry for the technical problem. The presentation at the very last moment got corrupted, but I hope now it's fine. Okay. I am going to give a presentation on Berestoid, in particular focus on what we have done in East Africa, rather. Next, it's moving. Next, okay. Okay, I think all of us, we know how, probably unlike to many other continents, African continent is very vulnerable to invasion, and this is due to so many factors. And of course, always the first response will be excessive use of insecticides, and it was also the case for the fall armyworm. So when fall armyworm it came, of course it came to find the environment is very suitable. There are so many host range there. And of course, as the previous presenter mentioned, it has high fecundity and high ability to disperse. And of course, when it comes to natural enemy, always when a new invader come, largely would come without natural enemy. But this is a yes and a no in case of the fall armyworm, and I explain further as we go ahead. So at ECB, as probably Subi mentioned, we have developed a comprehensive management for fall armyworm. That include biopesticide, which my colleague Dr. Subi elaborated very much on this one. And of course, we also optimize the trapping. And the good thing, by then we have a well-established boot boost system that was developed for management of stem borer, striga, and of course for soil health. And it happened when it was tested, it worked very well for this one. But here our focus on the breastoid. So there are some work that did by Mark Kenneth, and he realized that there are so many. The diversity of Africa is so high. Africa and other places where it's invaded, the diversity is very high. So we zero in to find out, and this could be largely this could be why the fall armyworm initially it was exploded, but after few times somehow it is stabilizing. We are not saying it's not a problem, it is still a problem, but it's not as it was initially when it came. And the breastoid could be one of the contributor to this stabilization. So we went ahead to find out what could be among this very high diversity, what could be the breastoid that form new association with fall armyworm, and we find several egg breastoid that Telonomus, which even though it was not native to Africa, it has been imported earlier. So it formed new association with fall armyworm, different species also of Trichogramma and Colonus also. They form that particular one is a egg larva. Three of them are egg breastoid, and one is a egg larval breastoid. And also there is a rich guild of the larval breastoid that also established new association with fall armyworm. Of a particular interest is Cotis-ACB. It's why we tested it was quite efficient, and this is why we took it for further investigation. So as I will show in my subsequent slide. So we took also this the egg larval breastoid and the egg breastoid. We tested them in that purely under lab condition. And their persisting range as from 45 for Trichogramma up to 90 for Telonomus. While for the larval breastoid, as I mentioned, the most effective among them is Cotis-ACB. And we did further tests to find out which particular host stage that is preferred for this particular species, that's Cotis-ACB. Of course, this for so many things. First of all, to optimize the mass rearing, because you don't want to offer the breastoid the wrong host, otherwise your colony will always collapse. And again, for the targeted releases, you want to find out when exactly should you do your releases. And interestingly, we found the early instars, those are the most preferred and most suited for this breastoid. When it comes to forcing star, It is not suitable. First of all, it defends itself against physically against the burstoid, and there is a very minimal cases of the encapsulation whereby the immune system of the host it encapsulates the egg or the larva of the burstoid. We also want to see when it comes to release of this burstoid, where could be the suitable areas. And as you can see from the model, around the equator, and as you go southward, it's quite suitable. And this under current climatic scenario, and this scenario remains the same even under future climatic scenario, with the slightly of the high suitable area shrink a bit. Having just to parasitize is something, but we went further again with this burstoid to find out the host species, because when you release, the burstoid will encounter so many other hosts. Some of them they even co-evolve with this burstoid. Like let's say Spodoptera luteralis, but there are other bees symbiotic in the system. Interestingly, we find this burstoid, it preferred. It is all the associated host that is Spodoptera luteralis, but almost equally same as fall armyworm, while the other hosts, the stem borers, they are not preferred. And of course, this translated to whether the burstoid is able to distinguish between the good burstoid or not, and those one which are not. And the suitability for this burstoid for opposition, it was high for fall armyworm, and its closely related relative is Spodoptera luteralis. And we went ahead and test how it affects the development duration, and number of cocoon form, and the number of closed wasp, and of course the sex ratio. And for all these parameters, the performance of this burstoid was highest in fall armyworm and Spodoptera luteralis, as evident from the table. Of course, except for the progeny, it is comparable across the host where the burstoid were able to develop. Then we went ahead, and now we establish all these parameters. We went ahead and do argumentative releases for several of these burstoids, the larval burstoid and the egg burstoid, and under different initiative. One of the project led by SUBI, another an EU and European Union funded project. We made releases all in so many countries in East and Southern Africa, and with a very promising result. But to make the impact more sustainable, we train private sectors, and in particular Dream Team Kenya, and they release over millions. And the farmers, they can access the burstoid from them at a very affordable price. Borrowing from what we did on fruit fly, where we do argumentation, where we test something called argumentorium. Argumentorium is like a tent. In this case, we use a small cage made of tent in such a way that the emerged larvae, or the hatch larvae rather, will not be able to exit from that netting cage, while the burstoid will be able to exit. So we will collect the larvae, put them in this particular cage in such a way that the burstoid, when they come, they drown in water. And we found out this approach it can increase the breastfeeding so low, while the eggs, the hatching larvae, the mortality is 100%. This, of course, we did it on a very small scale, like 1 acre, but just it's something to be taken into consideration. Then, having tested the burstoid with bush bull system, of course, we already the suppression trait of the companion crop, like this desmodium and the grasses, this is well established. But we also found that the volatile emitted by this plant, they recruit parasitoids that help further in suppression of this pest. And okay, with another initiative, we want to establish, okay, now we have released those parasitoids, how far they have gone and what will be their impact. So we release this parasitoid and we measure their dispersal from the central point, and just in one season, we found that they can disperse to more than 1 kilometer in a season with a precision of almost about 80%. This about the native natural animal or already which are in Africa, but okay, so now we have this parasitoid. The project come and go. We need to train the natural system of how to initiate and maintain those parasitoids, and we have done a very thorough training in Tanzania, in Uganda, and of course, as I mentioned, the plant village in Kenya. But being the invasive pest, of course, you agree with me, still the ideal target for the controlling it will be classical biological control. We are lucky IITA, they have already introduced 2 parasitoids. One of them is the egg parasitoid, and one of them is the larval parasitoid. So they share with us this material, and we test them in the lab. First of all, to be able to release them, we need to meet the requirement by our regulatory body in Kenya. One of them is to test their whether first of all they are effective against the follow armyworm, and whether also they were attacked as a related pest or other related species. For the egg parasitoid, that Colonus insularis, it was quite specific, and also it is quite efficient. For the larval parasitoid, that's Cortesia maglomeritilis, it has a slightly broader host range, but it is even more efficient than the egg parasitoid. And now jointly with CIMMYT, with a project funded with CIMMYT, which just has concluded, thanks to Barsana and his team for the resource. We released this parasitoid in 2 countries in Kenya as per the directive we are given from CAREF that we have to restrict for now our releases. So we released in 2 countries, that's Machako and Imbu. And of course, before we did the releases, we have a baseline information of parasitism. So the parasitism by other birds is quite low. And then we did after the assessment, after the first release, after the second release. And as you can see, the release is increasing, but still this is just preliminary. And the releases we have done, we need to continue. We have just done a few releases, but this one need to continue if we have to have an impactful suppression for fall armyworm. And I think there's so much we can do together. And with that, I want to stop here. Over to you.
All right,
Chairman.
thank you very much, Dr. Samir, for the presentation. So we're done with all of the 5 presentations, and I think our participants are very eager to ask questions. And in fact, some of them have already thrown in their questions in the Q&A chat box of Zoom, and some of our research speakers have actually started responding to them. But for an open forum for about 10 minutes, I now invite Dr. Prasanna to please kindly moderate our Q&A part. Thank you.
Thank you. I think, colleagues, you will agree with me that these are high-quality presentations on eco-friendly approaches to fall armyworm management. And many questions have come in, and which we are already responded to many of them, and some are being responded to by my colleagues. But if you have any specific relevant question that you would like to address or ask, please raise your hand, and we will try and answer that. Okay, go ahead, and colleagues may raise their hand. And then Sunny, please enable to unmute them so that they can ask the question.
We have a question from Mr. Hin.
Yeah, thank you so much. Can you hear me, everyone?
Yes, we can hear you.
Yeah, thank you, Mr. Moderator. It is so exciting to listen to different research results based on the study on fall armyworm. I have a question for Dr. Christine. Okay, so my question is about the presence of ants. What I heard from Dr. Christian is that ants are the best predator to deal with fall armyworms. So can they be bad for the crop as well? When we talk about ants, can they damage crop roots? Okay, thank you.
That's an excellent question. Yeah, that's an excellent question. And we are not talking of termites in this case. We are calling of real ants that forage around. And we've also found, interestingly, some of the legumes attracted some alternative prey, aphids, for example, which are not so good. We know that, but because aphids exudate some sugary solutions that attracted the ants, and then when the ants were there, they were starting to control the fall armyworm. So building on that, we put in some sugar solution as well in the fields to just test if that would make any difference if there was some sugar solution there. And indeed, that attracted again the ants, which then controlled the fall armyworm. So it's a bit complex because it's not a direct link. It's often indirect. What attracted the ants to come in and then see those fall armyworms and then control them on the sideline as well.
Subi, would like to add anything, Subi?
Yeah, I think Christian covered a lot. In our experience also, when we sprayed things like the fish soup, which is actually an animal protein, and ants goes for animal proteins, and specifically when they don't find, they start feeding on following. That's what we also realized as the management mechanism when you use fish soup, which very well relates to what Christian mentioned. Thank you.
Yeah.
Okay.
So can I have a follow-up question? Yeah, can I have a
Yes, go
follow-up
ahead.
question? Yeah, thank you so much, Dr. Christian, for your response. Actually, your research is basically on corn, but armyworms, I can say damage a lot of crops in Cambodia, especially rice. So can you tell us a little bit more about how can armyworm become an outbreak? What are the best factors to cause an outbreak of an armyworm? Okay.
Well, I must say this is beyond my knowledge level, and I could not give you a very good answer on that because I have not worked on rice in the past. But maybe Prasanna, you have some responses that are more elaborate than mine.
About what? What is that question?
How to prevent
I just want to know
fall
the
armyworm.
effect of armyworms on rice because it is the worst case in Cambodia. Thank you.
Okay. So far we have not noticed any major economic damage to rice or wheat because of fall armyworm. So laying of eggs on any crop plant is not an indication it is causing an economic damage. The economic damage is something that one need to very carefully monitor. But as of today, there are no such reports. But one has to clearly watch out for evolution of the insect strains. And if there is any new acquisition of capacity to cause damage, one needs to carefully monitor. But as of now, no.
Okay, thank you.
So can I actually have some?
Yeah, please.
So in our country, Bangladesh, in the Bangladesh Rice Research Institute, we did some this laboratory works on this fall armyworm on rice, and we have seen that fall armyworm can develop on rice. Maybe it takes some more times than the maize, but in the fields we are not getting any infestation. The main reason is behind that because rice is generally needs more water. And one of the lifestyles of these types of armyworm, like this Spodoptera litura or Spodoptera this progipada, this pupation is actually going for pupation underneath the soil. So as there is this muddy water, and due to that, we have seen even Spodoptera litura or Spodoptera fijupada cannot actually sustain in this rice field. So this is the main problem, but we are suspecting in some of the this upper land rice like this in our country, house varieties or something like that, where less water is using, might be a chance of infestations, more infestation of fall armyworm. Thank you.
Yes, thank you. I'm sorry that my question is out of the topic today, but I just don't want to focus on rice. I'm curious about the outbreak of fall armyworms. In case we talk about corn again, have you observed any specific factors or situations that can cause an outbreak of fall armyworms? Yeah. Even we talk about corn. Thank you.
So, Dr. Dr. Alam, have you seen any observation of fall armyworm in rice in Bangladesh?
No, not in Bangladesh, but there was some probably from Tamil Nadu. There was some infestation we have seen, but it's not so much. But in Bangladesh, not in the field. But as I told you, in the laboratory, we have seen that they can actually all the four this life cycle stages can be actually okay in rice. They can actually do it, but it takes some more times. So there is a chance. And as you know, there is two strains of this fall armyworm. One is rice strain. So if there is the rice strain can attack, so there may be also a chance of more infestation.
Okay. So
Yeah.
yes,
I just want to quickly add on to this. In this case, for a pest outbreak, there are a lot of factors that contributes to outbreak. Especially in an irrigated situation. Rice is an irrigated crop, and if in an irrigated situation you don't have anything else around, following a maize season or something, yes, the rice in that irrigated farm can get damaged by fall armyworm. This has been seen with maize as well. Irrigated maize is much more susceptible to fall armyworm as compared to any other because of their presence alone. So such kind of situations can happen, but wide reports on fall armyworm damaging rice has not come up so far, even in the presence of rice strains and the maize strains as well.
Yeah, thank you.
Okay, any other question, please? It appears most people are happy writing those questions on Q&A, and we are trying to respond as much as possible quickly. Okay, if there are no further questions, no raising of hands, thank you so much. Sunny, please go ahead.
Thank you very much, Dr. Prasanna, and to our resource speakers for graciously responding to the questions of our attendees. All right, now, so before we close, we just have some few more things. The evaluation form. We'd like to hear your feedback to our webinar. This is also our certificate request form, so kindly fill out the form, type in bit.ly/54solve, or kindly scan the QR code for you to access our feedback form for today's webinar. Okay, I think that's it.
Okay, with that, thank you so much. And once again, our heartfelt appreciation to the SEARCA team, including the director, Dr. Nova, as well as Sunny, and the entire team, which has put forward an excellent webinar structure together. and to enable us to reach out to all of you. And my sincere gratitude also to all my colleagues, the resource persons who have spared their time despite their schedules, especially colleagues from ISIPE, Dr. Subbi Savagan, who is always there. We have done perhaps this many, many times together. and Samira, who is another fantastic scientist. Both of them have always been there to support this kind of knowledge sharing events. And to my own colleague from Christian Terfield, he is an outstanding cropping system specialist and one who loves to work with the farmers. And that passion one can see in his presentation again today. And thank you once
Thank you
again,
so much.
Christian, for joining. And last but not the least, our very dear friend from Bangladesh, a very renowned entomologist, a former CIMMYT consultant and ex-director of Bangladesh Agriculture Research Institute, Dr. Syed Nurul Alam. Again, he is someone who is passionate about this field. And whenever we requested him to give any talk, he never even hesitated a second. So Dr. Alam, very, very grateful to you for always sparing your valuable time. And I'm sure that we can do a lot more in the years to come, thanks to your passion. So my participants, I can see more than 150, 60 participants, 160 participants, very actively engaging and asking questions. What is really important is don't consider this as a one-off event. You have our email details. SEARCA can provide them along with these presentations. If you have any follow-up action that needs to be done, whether it is with regard to access to different technologies or whether it is regard to any future possibilities of collaboration with organizations like CIMMYT or from like ISIPE, don't hesitate to reach out to us. SEARCA is a fantastic facilitator, and we can do many, many things together. I must also gratefully acknowledge the support from Melinda. Melinda Limlenko is my colleague who has formed this bridge between SEARCA and CIMMYT and enabled organization of this workshop. Many thanks, Melinda, in case you are not sleeping, but I know it is it's almost early hours in Mexico. Thank you so much, friends. We really enjoyed participating in it, and I'm sure SEARCA will follow up with many other follow-up actions. Once again, thanks a lot. Take care.
Thank
Thank
you,
you.
everyone.
Thank
Thank you to all our
Thank you.
attendees. And of course, to
Thank
our
you.
international experts, thanks everyone. And of course, on behalf of SEARCA, our sincere thanks to CIMMYT as well for this wonderful collaboration to bring fall armyworm management knowledge to our audience here in Southeast Asia and even beyond the region. And to our attendees, we hope to see you in the next webinar. So please stay tuned for announcements on SEARCA's social media channels. Again, my name is Sunny on behalf of SEARCA. Maraming salamat po. Thank you very much. And we wish everyone all a good day. Bye-bye.
Thank you.
Bye-bye.
Bye-bye.
Thanks, Dr. Prasanna.
Bye-bye.
Thank you.
Come, let's build the center that we'd like to be. A place where you and me can work in harmony, where everyone is proud to serve with dignity. Come, let's make this dream a reality. Fill our hearts and minds with our vision. Let our words below our own nation. Leave our values on excellence in work, commitment and unity. share a stream for the senior region, extended to the world throughout generations. Fill our hearts and minds with ambition. Side by side we stand for our mission. Leave our values on excellence in work, commitment and unity. Let's show the world we can. With God close at hand, we will grow together for the better.